Hello and welcome to the lecture 4 of paleontology. In this lecture, we will study about the Oparin Haldane concept of origin of life. As we have seen कि पहले कितनी सारी थ्योरीज आई बट उन्होंने प्रॉपर तरीके से ओरिजिन ऑफ लाइफ में कुछ हमें ऐसा टेंजिबल या ऐसा स्ट्रॉन्ग पॉइंट नहीं दिया जिससे हम ये समझ सकें कि ये लाइफ कैसे ओरिजिनेट हुई. तो लेट्स सी द न्यू मॉडल, द केमिकल और द मॉडर्न कंसेप्ट ऑफ ओरिजिन ऑफ लाइफ. So this theory was given by Oparin and Haldane. It is also known as CHEMOGENY or theory of origin of life. According to this theory, we can see that abiogenetic component like inorganic components have given rise to the molecules which readily developed into living cells. So the modern theory is also known as chemical theory or the theory of abiogenesis. In this theory, the hypothesis was that all the things, all the life which is coming, they are coming from the non-living material. The condition of the primitive earth was different from the present earth. As today we have oxygen on the earth, at that time oxygen was not there. It was like a different type of atmosphere totally which is today. So the idea was proposed by Oparin and Haldane, which have made the following assumptions. Spontaneous generation of life under the present environment condition is not possible. Early Earth's atmosphere was very different from the current environmental condition. Primitive Earth's atmosphere was reducing in nature right now today as it is oxidizing. Under these conditions, the chemical molecules, the inorganic molecule, जो भी inorganic components थे वो सब एक साथ react किए, फिर बार बार उनके reaction से कुछ organic biomolecules बने, फिर उन biomolecules का जो organic biomolecules बने उनका आपस में फिर reaction होता रहा, जिनसे complex type of cell structures जो आज हम देख रहे हैं इनका निर्माण हुआ और इनका पृथ्वी में ये आए. The solar system and the energy in the UV radiation provide the energy for the chemical reaction. Now what are the experimental evidences? जो हेल्डेन की कंसेप्ट था इसको बहुत ज़्यादा सपोर्ट नहीं मिला बिकॉज इस थ्योरी का कोई ऐसा अभी तक उस समय तक कोई ऐसा प्रैक्टिकल नहीं था. बट मिलर यूरे ने जो एक एक्सपेरिमेंट परफॉर्म किया, इट वाज अ कॉम्प्लेक्स एक्सपेरिमेंट परफॉर्म इन द लेबोरेटरी. इन्होंने एकदम वही अर्थ की कंडीशंस मिमिक की जो उस समय हमारी प्रोटो अर्थ की कंडीशन होती थी. So in this what they do, the experimental setup was consist of a glass flask, condenser, liquid flask was connected with tubes, and there was source of electric discharge which was providing energy. उस टाइम पे भी बहुत तेज हवा चलती थी, बहुत इलेक्ट्रिक डिस्चार्ज होता था, तो उसको उन्होंने एज अ फॉर्म ऑफ एनर्जी, एज अ यू कैन से लाइक इनिशियल फ्यूल की तरह यूज किया. देन he introduced a mixture of methane, ammonia and hydrogen in the ratio 2:2:1 and water vapor, and then he started heating it at 8000 degrees Celsius. Then what happened, slowly gradually this mixture was evaporating and condensing, and it was in a closed system for 18 days continuously. He provided energy in a form of spark, which was like the this electric discharge which we see in the atmosphere today. It was around 75,000 volts. The electric spark mimics lighting of the primitive Earth's atmosphere. Then the mixture was collected from the stop cork. वहाँ पे एक सैम्पलिंग कॉर्क था, उससे उस मिक्सचर को निकाला गया. फिर इस मिक्सचर को जब उन्होंने एनालाइज किया, देन इट इंडिकेटेड द प्रेजेंस ऑफ अमाइनो एसिड्स. बहुत सारे अमाइनो एसिड्स का जो है यस The many types of amino acids are found over here, such as glycine, alanine, aspartic acid, nitrogenous base like adenine, and simple sugar like ribose. Now, in addition to this, he also found hydrogen cyanide. Right? He also found hydrogen cyanide and formaldehyde like CH2O and other intermediate compounds as the acetylene and cyanoacetylene. As you can see over here, the carbon dioxide reacted with carbon monoxide and give you nascent oxygen, the breakdown of CO2 into carbon dioxide and then nascent oxygen. And methane is reacting with two molecules of oxygen, giving you formaldehyde and water. Then carbon monoxide reacted with ammonia to give you hydrogen cyanide and water. Then methane and ammonia reacted to give you hydrogen cyanide and hydrogen gas. Now from the glycine, the formaldehyde, ammonia and hydrogen cyanide reacted to give you glycine. Now this is how the glycine has formed, which is a complex type of compound. Then this is the setup of, let me show you. Yes. This is the complex set of Miller and Urey. Here you can see that in this flask they have taken lot of gases which are like water vapor, methane, ammonia, hydrogen, right? And then here you can see that they have taken the water which is like proto ocean, or you can say proto soup. Here they are heating it around 7,000 degrees Celsius, as I told you. Then slowly gradually it is evaporating, and again this is condensing over here. Then from here you can see the cold water is there which is letting this air to be condensed, and from here from the sampling probe the samples were taken out. Now what were the steps that were proposed in the origin of life? According to the chemical theory, in the origin of life, a series of chemical synthesis gave rise to the life. As per the hypothesis, the first one is formation of inorganic molecules. Now the high temperature in the primitive earth did not allow the condensation of the atoms into the inorganic form. So what slowly gradually, as the temperature was coming down, then all these components, all these abundant elements like hydrogen, oxygen, nitrogen, they were reacting with each other. With their reaction, complex type of inorganic compounds were formed like ammonia, methane, carbon dioxide and water vapor. The fuel for their reaction and their breakdown was given by sunlight and volcanic eruption. Then second, we have spontaneous formation of monomeric organic compounds. What are monomeric? Which have single identical units, like the simple molecule interacts with each other to form simple monomeric organic compounds. These compounds like fatty acids, glycerols, amino acids. Then we have organic bases like purines and pyrimidines, which will be definitely, as you know, will be used in the formation of DNA and RNA, right? The reaction between these inorganic compounds form simple organic molecules occurred in a reducing environment inside the ocean. So all these things were happening within that proto-ocean only. The organic molecule were condensed in the form of rain, and as temperature of the earth was lowered. तो जैसे जैसे ये टेम्परेचर लोअर होता रहा, ये जो आपका वाटर वेपर था, ये जो भी वाष्प ऊपर जा रही थी, ये कंडेंस होकर रेनफॉल के फॉर्म में फिर नीचे आई, फिर इनका एटमॉस्फेयर में भी और फिर उसी प्रोटो ओशन में दोबारा से रिएक्शन हुआ, तरह तरह के कंपोनेंट्स बनते रहे, जिससे धीरे धीरे एक प्रीमोर्डियल सूप तैयार हुआ, जिसमें हमने देखा धीरे से फिर ऑर्गेनिक कंपाउंड्स का भी डेवलपमेंट शुरू हो गया. As you can see in this diagram here, how the energy from the sun and then these all gases were reacting in the atmosphere, they condensed, then they gave rise to some organic molecules. Under the electric discharge energy was provided, some of the energy from the ultraviolet rays, you can say from the sunlight it is provided. तो फिर इन ऑर्गेनिज्म ने न्यूक्लिक एसिड्स का सिंथेसिस शुरू किया. न्यूक्लिक एसिड का सिंथेसिस के बाद हम अभी कंसेप्ट ऑफ कोएसरवेट्स की बात करेंगे. इन्होंने मेम्ब्रेन डेवलप करना शुरू किया अपने चारों ओर. उन्होंने एक आवरण, एक कवच या एक मेम्ब्रेन डेवलप किया ताकि इनका जो सेल कंटेंट है या इनका जो बायोलॉजिकल कंटेंट है वो सेव हो जाए. फ्रॉम हियर वी कैन सी दैट दिस इज द इनिशिएशन ऑफ लाइफ, राइट? दिस इज हाउ द प्री फॉर्म ऑफ द सेल कुड बी कंसिडर्ड. फिर यहाँ से डिफरेंट टाइप ऑफ डिफरेंशिएशन हुआ, जिसमें हमने देखा यूकारियोट्स, आर्किबैक्टीरिया एंड बैक्टीरिया. देन वी हैव प्रोकैरियोट्स, then we have a different type, like these are prokaryotes which have primitive nucleus and eukaryotes which have advanced nucleus, and near all the organelles are bound within nuclear boundary, right? So this is how slowly gradually the life came on the earth. Then the third step was spontaneous formation of complex organic compounds. The smaller simple organic compounds, see, they started reacting. Then with their reaction, the organic compounds like simple sugar, amino acids, polypeptide chain, like all these things were combined together to form a type of a cellular mass or cellular structure, या फिर हम ये कहें कि एक सेल के डिफरेंट टाइप ऑफ ऑर्गेनेल्स का डेवलपमेंट होना शुरू हो गया था. बट यहाँ पे अभी सेल्स में बहुत ज्यादा डिवीजन ऑफ लेबर नहीं आया था. दे वर सिंपल सेल विच वर जस्ट कैपेबल ऑफ जस्ट लिविंग एंड मल्टीप्लाइंग. देन वी हैव स्पॉन्टेनियस फॉर्मेशन ऑफ मॉलिक्यूलर एग्रीगेट्स. यहाँ पे हम एक कंसेप्ट की बात करेंगे, कोएसरवेट्स की. तो ये जो कोलाइडल एग्रीगेट्स है, ये आपस में कोलाइड करके दे स्टार्ट फ्यूजिंग टुगेदर. सो these are known as coecervates. This is a very important term. The layer of water molecule from around the protein molecule present in the coecervates. The membrane present around the molecule protect the molecule and bringing high concentration to enhance the chemical reaction. धीरे धीरे ये जो है कंडेंस होने लगा ताकि इनका कंसंट्रेशन बढ़ता जाए और इनमें रिएक्शन होता जाए. The colloidal aggregate absorb protein and other molecules from the ocean. और ये कैपेबल थे, यानी कि सेलेक्टिव परमेबिलिटी थी इनमें, ये एब्जॉर्ब करते थे प्रोटीन्स एंड अदर टाइप ऑफ सोर्सेज फ्रॉम द ओशन. So the result, then slowly gradually these coacervates starts growing in their complexities and in their organelle as well. The coacervates divided into multiple smaller ones. फिर इन्होंने division शुरू किया. Then these type of division as we see in the primary cells, अगर हम कभी प्रोकैरियोट्स की बात करें तो हमें इस तरह का बाइनरी फिजन कह लीजिए, मल्टीपल फिजन कह लीजिए, देखने को मिलता है. सो दीज coacervates with the initial species present in the ocean starts forming the primary cells. Then overall this whole story was done into two steps. First is formation of ebions or protocells. The coacervates has the ability to take up new molecules to replace the degraded molecule and maintain their size. Now as regeneration, growth and repair has started, धीरे धीरे इन्होंने अपने जो पुराने सिस्टम थे उसको छोड़ना शुरू किया और रीजनरेशन की पावर आई. They started maintaining their shape. Thus coacervates has a basic property of living system but they doesn't have complex molecule. अभी इनमें enzymes वगैरह हैं या फिर बहुत complex molecules develop नहीं हुए थे. The process of acquiring new molecule was not regulated. Later nucleic acids is entrapped with the coacervates and process of division became precise and controlled. Now they have a precise division and controlled division. That means somewhere that somehow the things are being engraved कि कैसे division करना है, कितना division करना है. This type of now this overall, if I call it, a primitive cell starts managing its own activities, right? The form of coacervates with nucleic acid is known as ebions. Now what are ebions? They are the coacervates which have nucleic acid within them. We can also call them protocells. Then we have formation of first cell. Protein molecules which have appearance of enzyme has enhanced the synthesis of several biomolecules in the ebions. फिर ebions molecule में क्या हुआ, प्रोटीन का डेवलपमेंट हुआ. एंड यू नो दैट एंजाइम्स आर प्रोटीन्स. सो स्लोली ग्रेजुअली उनमें एंजाइम्स का डेवलपमेंट शुरू हुआ. देन आरएनए एंड डीएनए डेवलप एंड दीज मॉलिक्यूल्स हैव टेकन ओवर द प्रोटीन सिंथेसिस. फिर प्रोटीन सिंथेसिस जो है वो आरएनए दैट इज राइबोन्यूक्लिक एसिड्स एंड डीएनए डीऑक्सीराइबोन्यूक्लिक एसिड, जिनको आप एज अ फॉर्म ऑफ हेरिडेटरी कैरियर भी कह सकते हैं. इनका इंटरेक्शन शुरू हुआ. इंटरेक्शन ऑफ लिपिड्स एंड प्रोटीन अलाउड द फॉर्मेशन ऑफ बायोमेम्ब्रेन which has provided selectively in the primitive cell for the intake of or exclusion of the material. Now the cell have started taking in food and excreting as well. It allowed appearance of the membrane bound protocells that has eventually given first cell on the earth. फिर इन्हीं ebions से या इन्हीं प्रोटोसेल से नए सेल जो डेवलप हुए उन्हीं को हम फर्स्ट सेल या फर्स्ट प्रिमिटिव सेल ऑफ द अर्थ के बारे में कह सकते हैं. स्लोली ग्रेजुअली डीएनए में म्यूटेशन होना शुरू हुआ और ये प्रीमोर्डियल लाइफ सेट हो चुकी थी, जिसको हम नियर अबाउट 2000 मिलियन ईयर्स अगो तक हमने ये देखा कि ये शुरू हुआ था. यूवी रेडिएशंस एंड इलेक्ट्रिक डिस्चार्ज, जो यूवी रेडिएशंस थे सन के या जो इलेक्ट्रिक डिस्चार्ज हमारे एटमॉस्फेयर में हो रहा था, ये इन्होंने इनिशिएट किया कि तरह तरह के ऑर्गेनिक मॉलिक्यूल आपस में इंटरैक्ट करें. इन द नाउ एट दैट सेम टाइम oxygen was also there in the atmosphere as it is today. Slowly gradually these molecules were condensing and the reaction was taking place after the development or the evolvement of oxygen in the earth's atmosphere. इससे पहले ऐसी conditions नहीं थी, इसलिए जो life थी वो बहुत ही primitive format में थी. Then we have gelatin. You can see here this is the complex type of compound. Then we have seen this is a change. This is a chain molecular reaction type of a thing. फिर स्लोली ग्रेजुअली टेम्परेचर ने इनको कंडेंस किया. कंडेंस होने के बाद एक मेम्ब्रेन बाउंड के अंदर ये कोएसरवेट फॉर्म हुए. कोएसरवेट के बाद इन्होंने कॉम्प्लेक्स स्ट्रक्चर्स डेवलप करना शुरू किया एंड देन दे गिव राइज टू प्रोटोबायोंट और यू कैन से दैट प्रोटोसेल. दिस इज अ प्रोटोसेल, राइट? सो दिस इज अ प्रोटोसेल. I must tell you that this protocell was different from the cell which we see today, but it was the initiation of the life on the earth. Then we have what are the some basic features of chemical evolution. The term evolution refers to the change from one form to another, right? Now we can also say here that it is a biological evolution because something which is living is evolving slowly gradually, changing in its composition, changing in its behavioral, anatomical as well as physiological structures. जो उसकी फिजियोलॉजी है, जो उसकी एनाटॉमी है, उसमें भी हम चेंजेस देख सकते हैं. The process of evolution can be understood from the fact that unicellular organism appears first simple multicellular and later, yeah definitely, as we see कि प्रोटोसेल बहुत ही सिंपल कॉम्प्लेक्स स्ट्रक्चर्स में डेवलप होना शुरू हुए. फिर आज देखिए कितनी कॉम्प्लेक्सिटीज बढ़ती चली गई और हम इवॉल्व होते चले गए. The fishes were the initial early vertebrates and it gradually changes to form amphibians. Yes, fishes थे amphibians. Slowly gradually हम इफ आई टेल यू दैट एक्वाटिक से टेरेस्ट्रियल हुए हैं, हमने फिशेस, देन फिशेस के बाद एम्फीबियंस व्हिच कैन dwell on land and water both, then we have different type of aves, mammals, and that keeps on going. The amphibians have produced reptiles that have evolved further and gave birds and mammals. Definitely फिर reptiles से birds और mammal बने. इस तरह का जो हायरार्की है ये हमने पढ़ रखा है और हम समझते हैं कि किस तरीके से डेवलपमेंट होना शुरू हुए. Then in the same series, mammals have evolved to humans and then ape-like primates by acquiring changes over the course of time. As you can see over here, from the Precambrian to Cenozoic, you can see how the life forms have changed, what are different type of structures that may have been developed. Some particular species were very dominant during some era, if I must say, like in Mesozoic, dinosaurs were dominant, in Paleozoic, fishes and all these reptiles or type of things were dominant, and in Cenozoic you will see that mammals are dominant, and definitely in Holocene you can see that man has the topmost priority, rather topmost in the overall evolutionary cycle. Thank you so much for watching my video. Thank you.